The biological control of boxing glove cactus is a spin-off of my official project, which is the biological control of a different weed, chain fruit cholla. Here you see the two weeds next to each other. They belong to the same cactus species, Cylindropuntia fulgida. There are two varieties of this cactus. The first one is my official project, Cylindropuntia fulgida variety fulgida, which is a tree cactus with very long, dense spines, looks white from a distance, whereas Cylindropuntia fulgida mamillata is in our country mostly represented by a very smallish cactus with crested cladodes. That is a chain fruit cholla with straight cladodes and normally a tall tree. This is in its natural home in Mexico. It also occurs in the southern states of USA. This is what Cylindropuntia fulgida mamillata looks like in its natural home when it is the normal plant. But this is what it looks like in South Africa. We have a crested form where some of the cladodes are flattened out like that, and that is what we call boxing glove cactus. The normal mamillata is not really boxing glove. Here are some more differences between the two species. You can see on chain fruit cholla the very long spines, long dense white spines, whereas on boxing glove the spines are much shorter and not as dense. They are also not barbed as far as I know, so they don't stick to you as much as the chain fruit cholla. Both species or both varieties have hanging fruits. In other words, fruits form a chain. The flower is produced on the tip of the first fruit, so the next fruit sits on the tip of the previous one. That's the color of the flower. I've never seen a flower on boxing glove, but there are fruits. In both cases, in South Africa, the fruit has no seeds. So the only way of reproduction is by means of cladodes that drop off the plant and root. There you can see it on chain fruit cholla, there you can see it on boxing glove cactus. So in both cases, they reproduce only vegetatively. Boxing glove cactus is still under the responsibility of the Early Detection and Rapid Response Program, and it happened because when people became aware that it is a problem in South Africa, nobody had any idea about its distribution in the country, and therefore it was an early detection target to find out where it's growing. And then at the same time, we'll get to that later. Infestations are mostly in Northern Cape. There are some very bad ones in Namibia. This is also in Northern Cape, but these three are in Northern Cape in a community called Heuningvlei, north of Kuruman, that was only recently detected, but there are about 400 hectares, very densely covered with many different cactus species, amongst which boxing glove is the worst one. You can see how it is encroaching on the gardens. Many of the homes have just been left standing because the cactus goes up to the door, and apparently the people just leave their houses and start a new one. Goats have a very difficult time there. The cladodes stick to their mouths and cause festering sores, and of course that's also a way in which the cactus gets dispersed. This is just a photo I received very recently from Sandy Lloyd in Australia of their worst infestation of the same weed. So this is really an enormous problem. Now, fortunately, we had, at the time when we realized that boxing glove was a problem, we had a potential biocontrol agent in the form of a Cochineal species that has been released against chain fruit cholla and was extremely effective. it was a specific biotype of the cochineal. We already have a different biotype of this cochineal in South Africa, which is controlling imbricate cactus, Cylindropuntia imbricata, and its origin is a third cactus species, Cylindropuntia cholla, in Mexico. So it's a new association. The cochineal wasn't collected from exactly the same weed as our target weed, or same plant species as our target weed. And in this case, there was apparently no resistance in the plant to the cactus, up to the cochineal, and therefore it did very well. The research of this was done by Helmut Zimmermann and Catherine Masenge, who now lives in Australia. She was a UCT student of John Hoffman's, and she did the host specificity of different biotypes and decided that this one, which we call, the cholla biotype, is the most effective one to kill chain fruit cholla. And because of the close relationship between the two cactuses, the chain fruit cholla and the boxing-clove, we predicted that the same cochineal would be effective in controlling boxing-clove cactus. This is just what the cochineal looks like. The females are covered by waxy substance. That is a female from which the wax has been removed. That's the egg that is produced by the female. These eggs hatch and form nymphs, and the first instar nymphs are called crawlers, and they are the stage that can disperse from one cactus plant to another one. The male nymphs or crawlers eventually produce something like a cocoon, and they, when they are mature, they have two wings. So the males can disperse and find females, whereas the females just sit on the same cactus plant for their whole lifespan and only produce eggs. It was first released against chain fruit cholla in 2008 and was extremely successful as far as cochineal biocontrol goes. It was a very fast success and also a total success in the sense that very little living plants were left. That's the release site in November 2008, and by June 2010 the same site had absolutely no sign of living cactus left. So this was on the original cactus chain fruit cholla. When we made a test release against it, against boxing-clove, I wasn't present. I sent the stuff off, and Sean Crozette and Trevor Shivuri were so good to release them for me. And after less than a year, all the plants there were looking like this. All the little cladodes lying on the ground were already attacked, and remember, the little cladodes are the only way in which this cactus disperses. Already the natural enemies of cochineal had found it, unfortunately. And there you can just get a view of how dense what big numbers of cochineal were present on the plants. And this is the right stage to distribute them, because there are lots of crawlers on this picture. We cannot expect this project to be without problems. Although I showed you for chain fruit cholla, we had some problems with the size of the plants, that they had very woody stems, and those woody stems were resistant to the effect of cochineal, and I'm expecting that the same thing might happen with boxing-clove cactus. Although the plants, here you can see chain fruit cholla plants in Douglas. This is the successful story where all the cladodes are falling down, most of them are lying on the ground, and there's cochineal on all of them. That's the success. But that's only for small plants. There you can see if the plants are bigger when they get attacked by cochineal, most of the cladodes lower down on the plant will drop, but during the rainy season most of the cochineal washes off the plant, and that gives the cactus opportunity to recover, and there you can see the white cladodes, those are newly produced cladodes. So this is what I expect might happen to boxing glove. We overcame this problem in chain fruit cholla by waiting until the plants were well infested with cochineal and then slashing them. Here you can see a slashed plant, that part has died. There was some regrowth, but that was in the beginning. On this graph you can see the light green part is everything that has died, and the dark green is what still remained alive, and you can see there's hardly anything alive 2 years after slashing. Whereas this plant, which was as much affected with cochineal as that one, but was not slashed, instead of dying back, it has only grown larger, and you can see that in the dark green sections. The dark green sections have become bigger and bigger. So unless you slash the plant, the effect of biocontrol will not be total. So in view of that, my recommendation would be that we follow an integrated strategy with boxing glove cactus. Wherever the plants are very big and have woody stems, I would recommend that you use the slash method. The most important thing to watch out for is that when you redistribute the biocontrol agent cochineal, that you collect it from exactly the right plant species. Because there is a lot of cochineal out there in the northern and eastern Cape. Some of it will be the right cochineal species, Dactylopius tomentosus, but it will be the wrong biotype because it might come from imbricate cactus. There is lots out there that we don't know where it's coming from. I am doing work on that, but Trevor, whenever you are redistributing, you must make sure that you only use cochineal that you know where it comes from. It's only the stuff that you collected at Askham, and you must keep track of where you've redistributed and only collect that stuff for further redistribution. And then, as I say, if the plants don't die, you can wait until they've got enough cochineal and then slash them, and they should then succumb. Okay, I'd like to acknowledge funding from Working for Water and research done by Catherine and Helmut, my assistant technic Joel Mabotja, and then also the EDRR team who have been very good in redistributing the cactus already. Thank you very much.
One quick question for Hildegard. I would appreciate question and not comment first.
No comments to the team. Hildegard and the EDRR program, I think it's quite important that from management side, and I'm not sure if you have this, around how we can work around this. He is quite aware of the challenge that we had in the Northern Cape. It would be nice to see Philip in terms of the planning for the pigment to know that we won't repeat the mistakes that we did. But it's quite an interesting project that can save us costs in terms of herbicide for this.
Yes. I'm not sure how we're going to go forward with this, but I will be attending the DWIP meeting in Kimberley, and hopefully all the role players will be there, and then we can decide whether the project stays with EDRR, goes to Working for Water, who's going to do mass rearing, probably Marietta and Boshoff in Heuningvlei. But yeah, we will need lots of cochineal for Heuningvlei, and there will have to be a mass rearing station somewhere, and I think let's discuss who will be in the best position to do the physical redistribution.
Okay, if that's not, thank you. Then we'll have our next speaker.